Okay, here we go. Welcome, everybody. Good afternoon. Thanks for joining us for the Lunchtime Lecture Series from the Botanical Research Institute of Texas and the Fort Worth Botanic Garden. I'm Dr. Brooke Best, Director of Research Programs, and I and my research colleagues host this lecture series, which takes place the first Tuesday of each month. Our next talk, before I forget, is going to be, if I can share this, now I lost my screen. Our next talk is going to be Tuesday, October 5th, and Dr. Cristobal Sanchez will present recent discoveries of new species in Mexico. Hold on, this is not working.
Nope, she's back. Here we go.
I'm back in. Okay. We were just talking about this. This is funny. I should not tempt fate this way.
I thought it was my chance to shine.
Oh, sorry, Jason. Okay. Okay, so viewers, today you'll remain muted throughout the lecture, but you can type your questions in the chat at any time, and we'll go over those at the end during the Q&A. So we do have two speakers today, so we're going to save all of our questions for a single Q&A session at the end. But if you'd like to test out the chat now, we'd love to hear where in the world you're watching from today. If you want to say the Brit building, go ahead and do that for those of you Brit staff. We are recording the lecture for our archives, and the recording will be available on our YouTube channel in a few weeks. And if needed, links to other resources mentioned by the speakers during the talk can be posted to the Brit webpage for this event. So if something happens and we get cut off, you can always reconnect with the original link on the event page. And if you experience lags in video or audio quality, I recommend you turn off your video first and then try closing other programs or apps you might have open. And if that doesn't help, you can always try exiting the talk and then re-entering the session again. We've turned off the waiting room, so you'll come right back in. Okay, so let's get started. It's my pleasure to introduce today's first speaker, Stacy Holt. So Stacy is a master's student in Dr. James Beck's Plant Systematics Lab at Wichita State University. His graduate research is focused on understanding the genomic composition of the invasive fern Salvinia molesta using molecular techniques in combination with the use of field-collected samples and herbarium specimens. His research interests include plant biodiversity, invasive species, and studying the effects of polyploidy on plant evolution. Stacy's talk today is What is Giant Salvinia? Plastid Phylogenomics of the Salvinia molesta invasion. All right, Stacy, we're ready for you to screen share.
Okay, thank you so much. Here, I'll get this pulled up. Does that look good?
Looks good.
Okay, thank you so much. Hello, everybody. I'm very excited to be here today. I just wanted to say thank you to you all as well as Britt for having us. And as she said, my name is Stacy, and I'll be talking about the graduate research I'm doing here at Wichita State and Dr. James Beck's lab. So I thought I'd start with this first image here. This is an example from Caddo Lake on the borders of Louisiana and Texas. And if we look at the eastern side of the lake, we can see everything looks pretty normal. There's a clear outline to the lake and the water looks good. But as we move westward, it almost looks like the land is kind of moving into the lake. And this is just showing an example of an invasive population of Salvinia molesta. So this slide is just highlighting that this is an invasive species that can even be seen from a satellite image. And I would say that's pretty frightening within itself. So if we look more at invasive populations of Salvinia molesta, we know that it's a freshwater aquatic fern. And when it invades a freshwater system, it can have many detrimental ecological effects and cover much of the water's surface. And some of these effects include reducing light as well as nutrient availability in the water, decreasing the available oxygen, and changing the flow of the water system. Now, it's also been shown that Salvinia can create good habitat for organisms that pass diseases along to humans as well. And so we can see how all of these can have cascading effects on the success of the ecology of one of these ecosystems. Now, if we get more familiar with Salvinia molesta, we know that, like I said, it's a free-floating aquatic fern. Now, it has a horizontal stem or rhizome that floats just below the water surface, and it produces a whorl of 3 leaves at each node. So the 2 emergent leaves are green and ovate in shape, and the 3rd leaf is highly modified to resemble and function somewhat similar to a root for this plant. Now, the sori are borne on a long receptacle on the underside of the plant, But when you break these plants at the nodes, it creates multiple individuals that continue to grow and proliferate. And so this is how it can spread so aggressively. And it's been noted that under favorable conditions, Salvinia can double its biomass in anywhere from 2 to 6 days. So this thing is just a beast. It's like a monster. This slide is just showing the 3 primary growth stages for Salvinia molesta, beginning with this primary invading form that's easy to break apart, and the internodes here are actually longer than the leaves. Now, when you enter the secondary form, the leaves begin to keel over and the internodes become shorter. And in this final growth stage, the plants begin to come compressed together as there's competition for space. And this gives it the potential to start creating this characteristic mat form. Now, I mentioned the ability for it to double its biomass earlier, but it's also been noted that during an initial invasion, Salvinia molesta growth rate can actually become exponential. And it has a potential to form these thick mats that pile on top of each other, and they can be up to 1 meter thick in the water column. And so this next photo is just highlighting this, and it's kind of a bad image quality, but we can see that there's actually a floating island of plants here that have established on top of this thick Salvinia mat. And so we're just highlighting that this was once a clear freshwater system that's now kind of been taken over. And I was going to use a different image for this photo. It was a researcher standing in the water of a Salvinia mat, and they had placed an actual brick on top of this mat, and the brick is just floating in the water because the Salvinia is just so thick. And so again, just kind of painting this picture of the scary invader. And so we know that Salvinia molesta has been described almost around the world, but it was first described in Zimbabwe. Oh, sorry. I'm trying to move the screens here. There we go. Sorry, I couldn't see for a second. Okay, so it was first described in Zimbabwe around 1972, where it had become an aggressive invader in one of the local lakes, Lake Kariba there. But they thought it was strange because they noticed Salvinia had a morphology that placed it among a group of South American species. Now, it was later found in the wild in southern Brazil, where it's now known to be native. And since then, it's been noted in about 22 countries around the world and has become an aggressive invader in many of these countries, including the United States. So the USDA listed Salvinia molesta as a noxious weed around 1983, but it wasn't actually found in the United States until 1995, originally in South Carolina. And although this initial invasion was successfully eradicated, it was later found in Los Angeles all the way across the country in 1998, a few years later. And it's now been observed in more than 24 watersheds across the United States, including the states of Texas, Louisiana, most of these shown on this map, Alabama, Georgia, and Florida. So we think the spread of Salvinia is due to a couple factors. And one is from cultivation in aquatic gardens or aquaria, and people just kind of haphazardly disposing of it in other water systems. And the second way we think it's being spread is through boats or boat trailers, as we see in this photo here. And unfortunately, people do still sell this as an ornamental aquatic. And so as I've shown kind of the frightening pictures of the satellite image as well as invasive populations, you can get this thing in the mail, which just adds kind of to the scary story of this invasive, as well as how it's being spread throughout the United States. Now, one of the more, or I guess we should move on to controlling giant salvinia. How can we control this thing? And so the current efforts of controlling giant salvinia include herbicide treatment, as well as physical removal and kind of grinding up the plant material and then taking it out of the water. And then finally, there's public education about not transporting it between water bodies. Now, while me and Dr. Beck were out actually during our sampling, we got to see some of this public education in action, you could say. We would go to lakes and we would see signs such as this. I don't know if you guys can see my cursor, but there would be giant salvinia signs, you know, beware. Also, we got to talk to a lot of people who were maybe from the area or just traveling, but 2 grown men in full waders going around campsites is a little suspicious. So we got to talk to a lot of people and kind of tell them about what we're doing. And we got to see a range of responses from people not knowing about this thing to kind of having done their own research on it and figuring out ways that they could stop its spread as well. So one of the more promising avenues of control is biological control and the introduction of this South American weevil, Critabagus salviniae. Now, the larvae of this weevil aggressively eat the leaf buds of this plant and it's been shown to control at least some local outbreaks of Salvinia molesta populations. Now, the tricky thing is getting the establishment or successful establishment of this weevil in these populations. And I know that we know that it depends on many environmental factors, but it seems the most important factors are temperature, nutrients, and the growth stages of Salvinia, which I kind of highlighted earlier. So I mentioned that the species reproduces using vegetative reproduction. And there's an abundance of evidence to show that this is the only way it reproduces. And I got to see an example of this actually when we came to Brit. When you open up some of these sporangia on the underside of the leaves, they're usually empty. They have no spores, or the spores are inviable. And so this slide is just showing what an abortive spore would look like compared to a viable spore. And this result is not surprising because we do know that Salvinia molesta is in fact a polyploid. And polyploidy in plants is very common, as all angiosperm genomes show a history of whole genome duplication, and about 95% of fern species have also undergone polyploidization. So it's been recognized that polyploidy has an influence on the evolution of many plant lineages, but the direct effects on the short and long-term evolutionary potential of a species are still not fully understood. We don't know everything. Although I have just highlighted one of the negative potential effects of polyploidy, which is obligatory asexual reproduction, mostly due to failures in meiosis. Now, because polyploidy is so widespread and common in angiosperm and fern species, it's thought that there has to be some advantage to this condition, and it has to be advantageous in some way for it to keep reoccurring. And some of the proposed benefits of polyploidy in plants include an increase in allelic diversity, which can just provide an abundance of alleles and maybe increase the rates of adaptation. Also, the presence of multiple alleles at a locus can mask deleterious mutations. And finally, duplicated genes can have the potential to develop novel functions which were not previously present within the genome. Now, this slide we can see a mitotic chromosome count done in the late 70s for Salvinia, and it's showing 45 different chromosomes. Now, the base number for Salvinia is 9, and so we know that this thing is a polyploid. And in fact, more specifically, it's a pentaploid. And if we can see on here, it has 9 groups of 3 chromosomes each and 9 groups of 2 chromosomes each. So Kuryachin hypothesized that giant Salvinia is the offspring of a hexaploid, which is contributing 9 sets of 3 chromosomes, and a tetraploid species contributing 9 sets of 2 chromosomes. And if that got a little wordy or complicated, here's a little simplified example. So we have this hypothesized tetraploid and hexaploid parent, each contributing reduced gametes, forming this hypothesized pentaploid. And so we think that Salvinia molesta is a hybrid. But what we're wondering is, if it is, what are the parents of this species? And this leads me to my 2 main research questions for this project. And these are asking what genomes are in the allopolyploid Salvinia molesta, and how many hybridization events have occurred. And so with this research project, we aim to identify the parental genomes within Salvinia molesta and determine if this species represents a single or multiple independently derived lineages. So now that we kind of know what the focus of this project is and what our questions are, we can meet some of the potential parents for Salvinia, and this is the rest of the genus. So the genus Salvinia is comprised of about 12 recognized species, and even though it was discovered in Africa, there's plenty of morphological evidence to show that it more closely identifies with American species. And so we can pretty confidently exclude the Eastern Hemisphere species on this list. I've kind of graded them out there. And what we're left with is American species, and I've put a few of the American species on the screen here. And within these species, we can now focus on a subset of these based on morphological characteristics. And that's because a closely related group of species within the Salvinia genus is referred to as the Salvinia auriculata complex. And so the feature of these apically joined hairs, or egg beater shaped hairs on the abaxial leaf surface, is actually an apomorphy that unites this complex. And based on this character, the members of the Salvinia auriculata complex are deemed to be the most likely parents for Salvinia molesta. Also, morphologically, a paper written by Mitchell in 1972 hypothesized that the parents for Salvinia molesta were in fact Salvinia herzogii and Salvinia biloba. So here's what we know cytogenically about this genus. And if we can recall, we think we're looking for a tetraploid and a hexaploid parent. And that would seem to exclude at least some of the members of this genus already from this list. So we've got this list of potential parental species, and the next challenge for this project would be getting material. So in the 2-year timeline of a master's degree at Wichita State, we can see how this would be a challenge because there's 4 species that are from the Eastern Hemisphere, including isolated places like Madagascar, and the rest of the species are in South America, primarily occurring in southern Brazil, where it's thought to be native. And so with this problem, I think we can all think of an answer of how we would solve this, and that's using herbarium specimens. And so that's what we did. We used herbarium specimens, and there's just about over 10,000 Salvinia specimens on the GBIF database. So we decided to use herbarium specimens, and we either personally visited or got loans from 5 different herbaria, taking tissue specimens and labeled data. Now, the goal with our in-person herbarium visits was to sample broadly across Salvinia genus with a focus on that Salvinia auriculata complex. And we also wanted to obtain a set of geographically and temporally diverse Salvinia molesta individuals, which is possible with herbarium specimens. So we also went into the field as part of a larger population genetic study that's designed to test if the Salvinia molesta invasion is in fact just a single clone. And so during this sampling, we took 600 specimens from 28 different populations in the states of Texas, Arkansas, and Louisiana. And we'll include at least 1 individual from each of these populations in our phylogenomic study. Now, if you guys notice in all of these photos, there's one thing missing, and that's Dr. Beck. Dr. Beck was taking all the pictures while we were doing all the hard work. No, I'm just kidding. Dr. Beck did a lot of hard work, and the sampling was fun, but he definitely helped a lot. So from our sampling, here's what we currently have. We have specimens from all but 2 species and members of the Salvinia auriculata complex, at least one of each of them. And so in this project, we're going to take a 2-pronged approach. In the first part, we're going to sequence whole chloroplast genomes in order to identify the maternal parent for Salvinia molesta, but we're also going to collect a nuclear HYPE-seq data set in hopes of identifying both parental species. So using this approach, this is what some of our phylogenies may look like. And we know the plastid genome, also referred to as the plastome, is inherited through maternal transmission from a single parental species. And so looking at this plastome phylogeny on the left, we can see the number as well as the phylogenetic position of plastomes present within Salvinia molesta will tell us the maternal parent and give us an initial estimate for how many times this species has been formed. Now, because we know nuclear genes are inherited from both parental species, the sequencing and analysis of these nuclear genes will be used to identify both parental species. And so looking at this nuclear phylogeny on the right, we can see the presence of alleles from both parental species within the offspring. So now I'll kind of move on to our methods for obtaining this data. And these were just examples. These aren't actually our phylogenies for this project. So once the tissue samples were collected, they were stored in silica and dried until DNA extraction and isolation. And we performed a standard CTAB DNA extraction using the 96-well plate method. Now, we were a little worried at first that the material wouldn't grind up as well because I mentioned those hydrophobic hairs on the leaf surface. But using this reciprocating saw bead beater that Dr. Beck had made for the lab, it turned out successful. And this is one of the coolest things in the Beck lab, besides the graduate students, of course. But this is a fun part about DNA extraction in Dr. Beck's lab. So here we can see kind of a simplified flow chart for our methodology. And we begin with herbarium and field sampling, as I mentioned, followed by DNA extraction. We will then perform Illumina library preparation, where a unique primer combination will be given to each sample so that when we send it off for sequencing, it'll have its own unique identifying barcode. We'll then utilize genome skimming to create a contig, which will allow us to sequence the entire chloroplast genome and hopefully identify the maternal parent for Salvinia within our chloroplast phylogeny. Now, the same Illumina libraries will also undergo nuclear hybridization sequence capture, where a set of 451 nuclear genes will be targeted to help us identify both parents for Salvinia molesta. So now the question becomes, well, can we get DNA sufficient for genomic work from these herbarium specimens? And unfortunately, I have to spoil it and say, yes, yeah, we can get sufficient DNA, or at least I hope I can convince you that we did. And so with this first figure, we see that we certainly get enough DNA. And this graph is just showing the DNA concentration versus the age of the specimen. And as we can see, there's no significant relationship here but we do get at least 10 nanograms per microliter from every sample despite the age of the specimen. Now in this next figure, we see that we can also make libraries from these DNA extractions, and I put library concentration on the y-axis with the age of the specimen again, and we see no clear relationship. And then finally, we have the number of reads mapped in the chloroplast genome versus the age of the specimen. And so these are just showing that we did, in fact, get adequate data from all of our specimens, or at least most of them, despite their age. And so now we're wondering, well, what does one of our phylogenies look like? What do these trees look like? And this is very exciting for me because this is the first, or one of the first trees that I got to make as part of the Beck lab. Now, when we first made this tree, Dr. Beck got really excited. We got the data back, and so he kind of did some editing here and there that I might have not seen yet. So I have one of these hanging up on my wall, but I don't know which one it actually is. But in this figure, we can see that we have, from our chloroplast data, a maximum likelihood tree that we built with 42 samples, and we rooted this with a Zola seen here at the bottom. Now, we've only included the 100% support bootstrap values just to show that there's good support for the major branches in this phylogeny. So the first thing we can take away here is that the species from the two hemispheres form highly supported and separate clades. And so this is already a little farther evidence showing that we can likely exclude the Eastern Hemisphere species from the possible parent list of Salvinia molesta. Now, if we take a closer look, it's hard to see, but I've highlighted that the Salvinia molesta actually forms its own distinct clade with no other species within it. And so what this is probably strongly suggesting is that the maternal parent for Salvinia molesta is either not within our sampled sequence data yet, or is perhaps an unknown species. Now you'll also notice the really short branch lengths within this clade, and this is indicating that there's low genetic diversity within the Salvinia molesta invasion worldwide. And it also suggests that because of these short branch lengths, that there was a single origin for Salvinia molesta, and it was only made one time. So here's what we've learned so far. We know that we can eliminate possibly 6 species from this list as maternal parents for Salvinia. And if we can recall, we have more DNA ready for library building. And I think just last week, I performed or made more libraries that include at least one new species from the Salvinia genus. But we also have more material on the way. And so Dr. Beck has formed a collaboration with Dr. Schwartzberg at the Federal University of Vicotia in Brazil. And he is a morphological expert on Salvinia. So he's got together a package of about 35 samples that represent 8 species within the genus. And we'll hopefully be getting those soon to perform DNA extraction, library preparation, and go through the workflow that I kind of highlighted earlier. And so with those, that'll really just bolster and increase our overall total sampling. And we can see that now we have at least one specimen from each of the species and a lot more samples in this highlighted Salvinia auriculata complex. And so from this pilot data set, or our initial data set, we've learned a few things. And what we know now is that the Eurasian and the American Salvinia are monophyletic groups, and that Salvinia molesta exhibits low chloroplast sequence diversity. And this is consistent with its vegetative reproduction. So this was not too unexpected. Excuse me. And finally, Salvinia molesta's maternal parent is either an unknown species or we just haven't sequenced it yet, which is a little anticlimactic, but hopefully we'll get more results as we move on with this project. And so with that, I'd just like to acknowledge my mentor, Dr. James Beck, as well as Aaron Siegel and Brittany Sutherland, who we saw in the field sampling photos that helped me get to this point and include me in this project. I'd like to thank the National Science Foundation, as well as the CPing Network, for providing support and funding, and the awesome Department of Biology here at Wichita State, the Kansas Genome Sequencing Corps for doing some of our sequencing, and then of course, all the herbaria that we visited and got loans from, including Britt. And I think that's about it.
Thank you, Stacy. That's great. Oh, wow. I wrote down a lot of questions, so hopefully there will be plenty of time at the end. We will, if those of you watching, if you want to write down your questions now in the chat, you certainly may, to kick those kind of off your plate and out of your head so you don't forget them. But in the meantime, we will introduce our second speaker and let him get started. So Our second speaker today is Dr. James Beck. Dr. Beck is an associate professor at Wichita State University studying plant systematics and biodiversity. He received his Bachelor's of Science from Eastern Kentucky University. What's the?
Kernels.
Kernels? Okay.
Yeah.
At first I thought you said turtles, which would have been funnier. Got that in 1999 and his PhD from Washington University in 2007. He's particularly interested in wide-ranging species, studying the effects of polyploidy and genetic diversity on range size. He also studies a wide range of organisms, from ferns to angiosperms. And his talk today is entitled Cytogeography and Niche Evolution in the Giant Goldenrod, Reconstructed from hundreds of herbarium specimens. All right, James. Let's see. Ready to screen share? Here we go. We see the coyote.
See the coyote. Okay, let me get that up there.
There we go.
Okay. All right. Let's see. Let me minimize this. Yeah. Okay. Can everyone hear me? Good. All right. So I'm very pleased to be with you today, and I'd like to thank Brooke for the invitation. For those of you who thought you were going to be seeing two talks on invasive species today, you're going to be a little disappointed. This is a talk on a native species. I'm just highlighting another project from my lab that also uses herbarium specimens. Specifically, we're going to be talking about this guy, the giant goldenrod. If you know native plants, I'm sure you've seen the giant goldenrod. It's one of the most common goldenrod species, particularly if you're in kind of a wetter area. Basically, if you encounter a goldenrod with tri-nerved leaves and a smooth stem, you're looking at gigantea. It's found all over Texas, again, particularly in wetter areas, and you can see it has this huge range. It's more than half of the United States anyway. But there's more complexity here. There's more than one chromosome number here. So Solidago gigantea is known as a diploid, a tetraploid, and a hexaploid. In other words, you can find individuals that have 2, 4, and 6 copies of the genome in their cells. Well, here's just a brief idea of how this happened. This is kind of like one of Stacy's figures. Normally what happens is diploids produce reduced gametes, which make another diploid organism. But sometimes this happens. We get the production of unreduced gametes. You put those together, and you now have a tetraploid. This process can go even farther. You can get a reduced gamete from a tetraploid here, and an unreduced gamete from a tetraploid. You put 2 and 4 together, and you get 6. So now we have a hexaploid. And again, all 3 of these cytotypes are known in Solidago gigantea. So what if this fact has something to do with the large range of this species? What if diploids do well in one area, tetraploids do well in a second area, and hexaploids do well in a third area? And when you combine all of these kind of ecological preferences together, you get a large range. In other words, what if we find tetraploids in one area, hexaploids in another area, and diploids in a third area? Another way of saying this is, are Solidago gigantea cytotypes geographically and ecologically different? This, by the way, was my master's student, Maria Martino's thesis work. So the basic question is, do we find cytotypes clustered across this range, or do you basically find all cytotypes everywhere, just kind of randomly distributed? So what we really need are a whole bunch of samples from across this range, and we need to know the cytotype of those specimens. Now, for this project, we actually did start in the field. We collected a small number of specimens for chromosome counts, and you'll understand why here in a few moments. And we made 11 new counts, and we did this so we could see how well a genetic tool called microsatellites could predict the cytotype of a specimen. And just real briefly, for those of you that don't know what microsatellites are, these are just places in the genome where we get this kind of stuttering base pairs. You can see this AT stutter here. And it's very easy mutationally to lose or gain copies of that repeat. So you can see there's 40 base pairs in this total chunk here because the repeat is 4 base pairs longer than this 36 base pair chunk down here. So there are two different alleles here. We can amplify the whole chunk with primers that are in kind of the normal regions on either side. We can fluorescently label one of those primers. We can run them out on a genotyper, and so we can see those alleles. So this trace here at the top would be an individual with the shorter allele. This bottom one would be an individual with the longer allele, and this middle one would be an individual with 2 alleles, with both alleles. In other words, a heterozygote. And this is why we want this information. So what you see here are 4 different traces from 4 different individuals at the same locus, at the same microsatellite locus. You can see this top individual only has 1 allele type, a size of 159. This could be a diploid, a tetraploid, or a hexaploid. In other words, it could be a diploid with 2 alleles, 159. It could be a tetraploid with 4 alleles, 159, or it could be a hexaploid with 6 alleles, all 159. This second individual has 3 alleles. I think you can understand why this can't be a diploid. Diploids can only hold 2 alleles, so this one has too many alleles to be a diploid, so it has to be a tetraploid or a hexaploid. The same for this individual that has 4 different alleles. It could be a tetraploid or a hexaploid. Finally, this individual down here, though, that has 5 different alleles could only be a hexaploid because tetraploids can only exhibit 4 different alleles. So, in other words, this information at this locus can help us diagnose the cytotype of an individual. And the idea is that if you look at enough loci, kind of the maximum number of alleles you see at any one of those loci will identify the chromosome number or the cytotype of that specimen. So here's our total sample set. We have 628 total samples, 11 of which were field collected, in which we also genotyped. Then we genotyped microsatellites for 246 herbarium specimens. And finally, we had a set of 371 chromosome counts done previously by our collaborator, John Semple. We already know cytotype of these, of course, so we don't need to do anything else with them. The one thing you might be asking me at this point is, how did you extract DNA from these 257 specimens quickly and economically? And if you saw Stacy's talk, you know how we did this. We did them 96 samples at a time. We added tissue to these racks with ball bearings. We pulverized them with this bead beater. You get nice powdered tissue. And then we did a C-tab extraction. And here you can see the various shades of extract you get when you work with specimens of kind of various ages and various qualities. This technique consistently produces plenty of DNA, around 20 to 30 nanograms per microliter, which is way more than you need for microsatellites. By the way, this whole extraction is the subject of a video that's available on YouTube. If any of you guys need the link to that video, you can feel free to email me. Brooke can put you in contact with me, and I can send you that video protocol. So here's where we stand. This is what our sampling looks like geographically. The dark pink samples are the previously done chromosome counts. Those are the ones that over 370 chromosome counts. We know what those cytotypes are, so we don't care about those at the moment. We can put those aside. The dark green ones are the unknowns. We only have genetic data for them. The key here are the light green and the light pink samples. These are specimens for which we have both genetic data and chromosome counts. What we can do here is see how well the genetic data predicts cytotype in these specimens, to see if our genetic data in these dark green samples can be used to predict cytotype. Here's how this played out in a couple of example specimens. So here's an example. We have a chromosome count. We see images from this same chromosome count up here at the top. These show a pollen mother cell going through meiosis. And all three show that this specimen has 9 pairs of chromosomes, 18 total. So this thing is a diploid. Below we see the number of alleles that we see at 12 microsatellite loci. So at the first locus, we see 2 alleles, 164 and 168. At this third locus, we only see 1 allele, and so on and so on. As you can see, allele number is either 1 or 2. It doesn't exceed 2 at any one of these loci. So using only the information from the loci themselves, we would predict that this is a diploid. And we know from the chromosome count that it is indeed a diploid. Here's a second example. This thing has 27 pairs of chromosomes, 54 total. In other words, it's a hexaploid. And over here again, we see the arrays of alleles we see at 12 microsatellite loci. And the ones that I've kind of put in bold and black here, these are ones where we see 5 or 6 alleles, which excludes both diploids and tetraploids. So there's evidence at 1, 2, 3, 4, 5 of these loci that this thing has to be a hexaploid, and indeed it is. So we did this sort of thing in 20 samples. We had 20 samples for which we knew the chromosome number from a count, and we had genetic data. And we were correct 17 out of 20 times. 17 out of 20 times, the genetic data predicted the chromosome number. So nearly 90% of the time, we can correctly predict cytotype in a specimen with genetic data alone. 17 out of 20 times we were correct, so nearly 90% of the time. So in other words, what we can do is we can go back to these dark green samples for which we don't know chromosome count, but we do know genetic data, and we can predict the cytotype of them. So now we either know or we can predict cytotype from every dot on this map. And what I'm going to show you now is the same map, but colored by cytotype. So here's what we get. And as you can see, there's a clear pattern here. So diploids are concentrated kind of along in New England, the Atlantic coast, and in the southern United States. Hexaploids are in the western part of the range on the Great Plains, and tetraploids are basically in between. So you can see there's a clear geographic kind of non-randomness to where you find cytotypes in Solidago gigantea. And you can do all kinds of statistical comparisons based on latitude and longitude and show statistically that that each cytotype has different latitude and longitudes, mean latitude and longitudes. So you can demonstrate this pattern statistically. So there's this clear geographic signal here. This strongly suggests that these cytotypes are kind of differently adapted to different portions of the range. But we need to formally test this. The good thing is there's publicly available data for which we can get climate information for all 628 locations. And if any of you guys have ever done any kind of niche work, you've probably used this bioclim data. It's a set of 19 climate variables. Basically, it's precipitation and temperature kind of sliced and diced in different ways. First thing we had to do is get rid of a bunch of pairs, or at least a member of pairs of variables that were highly correlated with one another. So once we do that, we're left with these 8 climatic variables. So these were the data. We had these 8 climatic variables at all 628 locations. Then we can do 2 comparisons. We can, in the cartoon earlier, I showed you that diploids lead to tetraploids. So we can ask in the transition between diploids and tetraploids, do we see a transition in niche? In other words, are the niches of tetraploids different than the niches of diploids? Secondly, I showed you that tetraploids typically lead to hexaploids. So we can do a second comparison and ask, do hexaploids have a niche that differ from the tetraploid niche? So we have 2 kind of evolutionary transitions here, so we can do 2 comparisons. Let's start with the diploid to tetraploid transition. The first thing we did was a traditional principal component analysis on these ecological data for diploids and tetraploids. Then we could ask if the scores on the principal components differed between diploids and tetraploids. And they do, at least for the first 4 principal components. So scores of diploids and tetraploids differ on the first principal component and the 4th principal component. And if you combine all the PCs together, they differ statistically at the first 4 combined. So this is a very basic statistical way of asking if these niches are different, and they are statistically. There's a second way we can do this where we project each ecological niche onto graphical space and ask a couple questions. Here's what the diploid in black and the tetraploid in blue niches look like in kind of graphical format. Where they overlap is in purple. And we can ask a few questions statistically with this approach. We can first ask, are the 2 niches equivalent? Are they statistically the same? Also, since the tetraploid niche derived from the diploid niche, we can ask how much the tetraploid niche has expanded on the diploid niche and how much of the diploid niche has the tetraploid niche left behind. And we see again, no, these 2 niches are not the same. The niche equivalency test shows that the diploid and tetraploid niches are not equivalent. But this is subtle. The other approaches to look at this shows that the tetraploid niche has expanded on the diploid niche by only 4% and has left only 6% of the diploid niche unfilled. Another way of saying that, it's left only 6% of the diploid niche behind. The tetraploid and the hexaploid, or the tetraploid and the diploid niche differ from one another. But this is subtle, right? We're talking about single-digit kind of niche transitions here. So there's real but subtle ecological differentiation between diploid and tetraploid. But in other words, between black and blue here. And that's not so surprising if you look at the map, because there's a lot of overlap between black and blue here. But let's look at this second transition, that between the tetraploid and the hexaploid niche. Again, we see statistical evidence, basic traditional statistical evidence for these niches being different. They differ at 3 of the 4 principal components and at all 4 principal components if analyzed simultaneously. So we already have basic statistical evidence that these 2 niches are not the same. And if we do that second comparison where we look at them in graphical space and run a variety of tests on them, we can see that again, they're not equivalent. The tetraploid and hexaploid niches are not equivalent. But this time, there's been a far more drastic transition between these 2 niches. The hexaploid niche has expanded on the tetraploid niche by nearly 30%, by 27%, and left over 20% of the tetraploid niche unfilled. So in other words, we have a much more substantial niche shift in the transition from the tetraploid niche to the hexaploid niche. And this makes sense geographically. The hexaploid niche and the tetraploid niche overlap relatively little geographically. There's not that much overlap between red and blue here. To summarize, we see niche transitions for both cytotype transitions, but the one from tetraploid to hexaploid has been much more substantial. And these ecological transitions are likely partially responsible for the large geographic range of the giant goldenrod. In other words, this species has probably achieved and maintains this large range due to kind of the differential success of these 3 cytotypes in different areas. And so in this way, polyploidy is a major contributor to range size in Solidago gigantea. And there are many such species like this. There's many autopolyploid series like this, where under one species you have lots of different cytotypes. So we think that we've demonstrated this in another species in buffalo grass. We think this could be a common theme in autopolyploids, is that they achieve their ranges by cobbling together the ranges of the cytotypes. And this insight was brought to us, again, by genetic data that we got from herbarium specimens, specifically all of the dark green dots you see here. So without those dark green dots, we would have had a much poorer estimate of cytotype distribution in this species. I need to thank Maria Martino. This was her master's thesis. John Semple was who we got all of those 370 chromosome counts from, some funding from Wichita State. But most importantly, as always, we need to thank the curators and collectors of these herbaria. These are the 21 herbaria from which we sampled for this project. And I'd like to highlight 70 specimens that we sampled at BRIT, at VDB SMU BRIT. So we got 70 out of our, 260 novel herbarium specimens from BRIT. So big thanks to BRIT. And with that, I'm done. So I will stop sharing my screen. And there we go.
All right.